Welcome to video 2 covering NCBI Blast and Pairwise Blast. In the last video, we went over navigating NCBI to find gene and protein pages for GAPDH and Drosophila melanogaster. In this video, we will cover utilizing the blast function for the same gene. Blasting is an invaluable tool that is a staple in the annotation process. It is recommended that you bookmark this page for correct reference, as I have done. So what is BLAST? This stands for Basic Local Alignment Search Tool. BLAST finds regions of similarity between nucleotide or protein sequences to other sequences in the NCBI database. It then calculates the statistical significance, generates alignments, and allows us to examine differences in the domains. By comparing sequences in this way, we can determine the function of a sequence if it is unknown, or determine how well a sequence with a known function compares to its orthologs. I will now go over the types of blasts. Blasts can be performed on nucleotide or protein sequences. In the top left corner, there are hyperlinks to all the different types of blasts that can be conducted. Clicking on them takes you to the appropriate type, but be sure that you are using the correct sequence with each one. If a peptide sequence is needed for the input, make sure you use that, or nucleotide if applicable. So now to briefly explain the different types of blasts and their functions, I have this figure here. Blast P and Blast N are the most straightforward of these blast algorithms. Blast P takes an inputted peptide query sequence and searches the database for other matching peptide sequences. Blast N does the same thing, but instead uses DNA to search for DNA. Because of the way that open reading frames work, and you can watch that explanation in one of the previous videos that I did for the annotation worksheets, BlastX takes this into account by taking a DNA query sequence and translates it into 6 possible protein sequences using all 6 possible reading frames, and then uses those peptides to search the database for other peptides. And TBlastN does the reverse, in that it takes DNA sequences in a database, translates those into proteins, and then compares those against the protein sequence that you enter in as your query. Lastly is TBlastX, which is the most computationally intensive blast algorithm, and it takes DNA from both a query and a database into 6 potential proteins, and then performs 36 protein-protein database searches. And of these 6 programs, the most commonly used in NCBI on your day-to-day is going to be Blast P, and that's the one I recommend bookmarking just for quick access. But now to go into blast inputs and to show you how you would conduct a blast. Here is the page of where you would enter, and there are a variety of features here, but I will just be covering the basics of what I tend to use. The first thing to consider is the query entry. And there are 3 valid inputs when you're entering your query for the search. You can use the FASTA sequence, which is the peptide sequence, but it has that little caret at the beginning. Or you could just omit the caret and format and just put in the bare peptide sequence or nucleotide if you're doing cDNA. And lastly, you can just use the accession number. Though, just to make a quick note, you have to have the accession number entered exactly as it appears in NCBI. Like if there's underscores, those need to be included in the exact way they're found there, or else it won't work. So be sure to enter the accession number correctly. Most commonly, I tend to just use the FASTA sequence, but occasionally, if I just have the accession number, I'll use that. But it's important to note that both are valid. And an alternative to entering the query in the search box, it's possible to upload a text file with a query sequence if that's your preference. Another thing that you can do that I'll point out is that if you have a longer sequence of nucleotides or peptides, you can use these query subrange parameter boxes to limit the search, the blast search, just to a certain desired range within that sequence. So if you had an 1,000 nucleotide cDNA sequence and you wanted to just look at the 300-400 range, you could enter that and just limit the search to that. So these query sequences can be screened against the entirety of the NCBI database, or they can be filtered to only screen against certain organisms in the database. For example, the Insecta taxid is used if a D-Citry sequence is the query entry, though you can also use Hemiptera to search for hemipterans, and so the Insecta will search just for insects and hemipterans just for hemipterans. Similarly to how you would do this with an advanced search in NCBI, make sure that you select the taxid ID number from the dropdown, or else the search may not work properly. All other settings I tend to leave as defaults, and once all inputs are entered, clicking the blast button will proc the search. So now that we have run our blast and it has finished processing, I will now go over the blast results page. This is what it looks like, and before I begin interpreting the results, I want to point out a number of items. Blast results do not save if you copy the hyperlink. They will also eventually expire if you have them open in a tab, the time of which is listed here. If you will be frequently referring back to a blast page, it's possible to save a blast result if you have an account with NCBI, but typically if I run a blast and it's important to my work, I'll tend to just take screenshots of the descriptions, graphic summaries, domains, and sometimes applicable alignments, and put those into my notes. You can also save blasts as a text file or other formats by clicking the drop-down arrow next to Download All and selecting the desired format. The current version of Blast displays the results broken into these tabs. If you prefer to view the results holistically, all on one page, you may select Back to Traditional Results page. The summary box displays some general information about the search. Of note for quick reference is the query accession number, name, and length. The results in the description page show the hits, which are sequences in the NCBI database that match to the query. If you desire to filter the results by any of the listed parameters, for example by organism, you can enter them into the filter results box to narrow the results. The information provided in this video is not extensive, and there are many features that are not explained. For a more detailed description of this page, if there's anything that you're curious about, select How to Read This Report for a file that explains some more details. I will now go over interpreting the Descriptions tab. The description here states the names of the proteins or nucleotide sequences being aligned. The query coverage denotes what percentage of the query sequence aligned to a sequence in the database. The E value, or expect value, is a measure of statistical significance for the search result. The closer a value is to 0, the more significant your match is. Percent identity denotes how identical the query sequence is to the hit. And the score is a rating of quality of the hits. A general rule of thumb is that you want a high score with a low E value, and also a high percent identity and query coverage. But there are situations where a model could be improved, but those numbers could be lower. Situations like that, you'll have to kind of play as they come up. You can see here, the accession number is also provided. So I will now go over the graphic summary. The graphic summary is a page that displays the query sequence length up at the top in teal, with a ruler that labels how many units long the query is, if it's either a nucleotide or peptide sequence. Each of these bars in the graphic summary corresponds to a single hit that you can refer back to on the descriptions page. And by hovering over a single bar, it will display the organism that it comes from. The color of the bar denotes its score, with a higher score being more desirable. A color key is present at the top. The length of the bar shows where the sequences align. The last thing to note is the conserved domains up at the top. Domains were briefly mentioned in the previous video, but to explain them in a bit more depth, a domain is a conserved individual folding unit that makes up part of a protein. They are tertiary structures that can evolve, function, and exist independently of the rest of the protein. One domain may appear in a variety of different proteins, and comparing the domains that appear in the query to the hits is a useful tool. Using GapDH as an example, it is a highly conserved gene which has great query coverage and identity with other orthologs. A note about this graphic summary is how the red bars align to the query. Here, for example, with GapDH, our peptide is about 350 amino acids long, and we see these red bars aligning to it. Each of these hits is only going to show the region that it aligns to our query. But if any of these red bars were longer, so if an ortholog had a sequence that was maybe 400 amino acids long, but maybe there was some extra on one end, you couldn't see that in the graphic summary because the length of the graphic summary is limited to the length of the query. So it's possible that some of these sequences that have red bars are longer, especially if you see perfectly straight lines on either side of the edges of this graphic summary. So it's important to take a look at these different hits and ensure that our query sequence doesn't have missing amino acids or nucleotides if it's a cDNA sequence. Now to touch on alignments. By clicking on one of the bars in the graphic summary page, it'll send you to the alignment of that hit to the query. The alignments show a side-by-side comparison of each amino acid or nucleotide in a BLAST result. By default, it is displayed as pairwise, but there are other possible options to select for different displays. One that I like and tend to use is pairwise with dots for identities, which highlights in red any differences between the query and the hit. It's helpful to look at alignments because you can do things like observe the length of the matching sequence and observe where regions of similarity are. And it can also be used to identify gaps that may be found along the length of the alignment. So that's regular BLAST, but you can also conduct pairwise blasts. Rather than blast a sequence against the NCBI database, if you have two or more sequences that you want to examine and compare to each other, you can conduct a pairwise blast on them. To do so, you select the checkbox that says align two or more sequences on the BLAST input page. Then you enter the subject sequences in the new search box. Of note is you can use multiple different subjects and compare them against the single query, and that's a very useful thing to do. Of note is that if there is a discrepancy in the size of the two sequences, when the results of the blast are generated, issues could arise regarding query coverage. This occurs if the query entry in the top box is shorter than the one in the bottom box. If the shorter sequence in the top box was fully contained within the longer sequence in the bottom box, the results would show a 100% query coverage. This is potentially misleading, as if the positions of the sequences in the search boxes is reversed, it would show that the query coverage is less. as there is a segment in the logger sequence not present in the shorter sequence. Besides this, the results of a pairwise blast are the same as a regular blast, with only the inputted sequences being compared, as opposed to the entire NCBI database being searched. So this has been my tutorial on NCBI Blast. Next time we'll have you learning about Apollo and getting into the program and doing annotations and validations. Thanks for watching.